So good day everyone. Today I will discuss the RNA translation, also known as protein synthesis. So protein synthesis is the process by which cells make proteins. So proteins are essentials because they help to build the body structures and acts as enzymes. It also transports substances and perform many other functions. So translations is the second major step of gene expression. So in simple terms, it is the process of converting the genetic message carried by the messenger RNA or mRNA into a protein. So throughout this presentation, we will explore how translation works. So the molecules involved is its energy requirements and its importance in medicine. So now let's talk about termination in disassembly. So translation ends during termination. So this occurs when the ribosome encounters a stop codon such as UAA, UAG, or UGA. So unlike regular codons, stop codons do not code for an amino acid. So instead, it releases factors that binds to the ribosomes and signals the end of protein synthesis. The completed proteins is released and the ribosomal subunit separates for future use. And now translation begins with a stage called initiation. So in here, think of initiation as preparing a factory before production start. So first, the small ribosomal subunit attaches to the mRNA. Next, a special transfer RNA or tRNA is carrying the amino acid. Methionine recognizes the start codon, which is usually AUG. So after this recognition, the large ribosomal subunit joins the complex. So once all components are assembled correctly, protein production can begin. So to understand translation better, we need to look at the flow of genetic information. So DNA stores genetic information inside the nucleus. So during transcription, DNA is copied into mRNA. So the messenger RNA travels to the ribosome where translation occurs. Then finally, the information encoded in the mRNA is converted into a sequence of amino acid. So now it forms a protein. So a simple way to remember this process is DNA proceeds to RNA that proceeds to protein. For the next one is the ribosome workbench. So the ribosome is the site where proteins are made. So its function is like a workbench in a factory where amino acids are assembled. So ribosomes are made of ribosomal RNA and proteins. So they contain specific sites called the A sites and P sites, and also the E sites. So these sites helps to guide the transfer RNA molecules as they deliver amino acids during protein synthesis. So without ribosomes, translation cannot occur. So now before translation can proceed, transfer RNA molecules must be loaded with the correct amino acid. So this process is called the tRNA charging. So an enzyme known as amino acyl tRNA synthetase attaches the correct amino acid to its corresponding tRNA. So think of it as labeling delivery trucks with the correct package before the shipment. So this step ensures accuracy before the wrong amino acid could produce a defective protein. So after initiation comes elongation. So this is the stage where the protein chains grow longer. So first, a charged tRNA enters the A site of the ribosome. Then a peptide bond forms between amino acid. Next is the ribosome moves along the mRNA and the growing protein chain shifts to the next position. So this cycle repeats many times until the complete protein is formed. Now, translation requires energy. So cell use molecules called ATP and GTP as energy sources. So energy is needed for charging tRNA, so assembling ribosomal components and moving the ribosome along the mRNA. So although translation consumes a significant amount of energy, it is necessary because proteins are vital for cell survival and function. Cells have quality control system, so it prevents errors during translation. So these mechanisms identify damaged or faulty mRNA molecules. So its errors are detected the defective RNA is destroyed before it can produce abnormal proteins. So this quality control process helps maintain healthy cellular function and reduces the risk of disease. Some antibiotics work by targeting bacterial translation. They bind to bacterial ribosome and interfere with protein synthesis. So without proteins, bacteria cannot grow or reproduce. Examples that include is the tetracycline, erythromycin, and chloramphenicol. So these drugs are effective because bacterial ribosomes are different from human ribosomes. So ribosomopathies are diseases that caused by defects in ribosomes or ribosomal proteins. So when ribosomes do not function properly, protein synthesis become impaired. So this can affect the cell growth and development. Like for example, it includes Diamond-Blackfan anemia and Treacher-Collins syndrome. So studying this disorder helps scientists to understand the importance of proper translation. So in summary, translation is the process of converting genetic information from mRNA into proteins. The process involves initiation, elongation, and termination. And it requires ribosomes, tRNA, amino acids, and energy. So translation is essential for life and has important clinical application, including antibiotic therapy and the study of genetic diseases. So, thank you for listening. And that's all. Thank you.